Photosynthesis is an anabolic process essential to autotrophs as they become self-reliant for food sources. It is the process by which plants use light energy to convert water and carbon dioxide into glucose and oxygen. This process begins in the leaves of plants. The chloroplasts are like the photosynthesis factory, and the thylakoids inside are where light reactions specifically occur. The fluid-filled area outside of the thylakoid membrane is called the stroma, and the area inside is called the lumen. Important components of this process are photosystem II, the cytochrome complex, photosystem I, and the essential enzymes NADP plus reductase and ATP synthase. Now let's begin the electron transport chain. Photosystem II has two components. First is the antenna complex, which contains multiple pigments, and second is the reaction center. The antenna complex will absorb a photon of light, which will bounce off its multiple pigments to lose energy. When at the specific wavelength of 680 nanometers, the photon will enter the reaction center. It will then excite an electron of P680, which is the reaction center's chlorophyll A molecule. It is then denoted as P680 star. The electron will be stolen by the primary electron acceptor, causing the pigment to be oxidized. It will now be denoted as P680+ due to its positive charge. The photosystem now needs to become neutral again, so it must gain electrons, also known as becoming reduced. This is facilitated by an enzyme in photosystem II called the oxygen evolving complex. It breaks the water molecule and steals its electrons. The breaking of the water molecule releases 2 protons to the lumen. It will pass electrons to P680+, turning it neutral again. This process also releases an oxygen atom. 2 molecules of water must be broken in order to release an oxygen molecule to the environment. Throughout the ETC, many reactions involve the transfer of electrons from one species to another. The species losing electrons is oxidized, gaining is reduced. Together, these form a redox reaction. Then, the primary electron acceptor gives the electron to mobile carrier plastoquinone. Plastoquinone will move towards the cytochrome complex. It is then oxidized, giving up its electrons to the cytochrome complex, yet another redox reaction, and in the process, it moved a proton from the stroma to the lumen. The cytochrome complex is then oxidized and transfers its electron to plastocyanin. It then arrives at photosystem I, but it runs into a problem. Photosystem I is neutral right now, so it cannot accept plastocyanin's electron. To solve this, it will absorb a photon of light, which will first enter its antenna complex, a series of pigments, and when the light reaches 700 nanometers, the photon will be absorbed by the reaction center. It will then excite an electron of P700, which will then be denoted as P700 star. The electron will be stolen by the primary electron acceptor, giving P700 a positive charge denoted as P700+. It can now act as an electron acceptor and is reduced back to P700 by the oxidation of plastocyanin. The primary electron acceptor transfers an electron to ferredoxin, which is an iron-sulfur protein, and the final electron carrier. Ferredoxin will transfer the electrons to NADP plus reductase. The oxidation of ferredoxin results in the transfer of high-energy electrons to NADP plus. 2 electrons must be transferred, and a proton must be added from the stroma. This creates NADPH. Throughout the electron transport chain, a proton gradient has been formed across the thylakoid membrane. The lumen is highly concentrated with protons compared to the stroma. This happens first because 2 protons enter the lumen per each water molecule that is split. Second, 1 proton is moved to the lumen each time plastoquinone is reduced and oxidized. And finally, NADP plus reductase removed a proton from the stroma per NADPH molecule made. The higher concentration of protons inside the lumen results in a lot of potential energy, generating a proton motive force that drives protons back into the stroma down their electrochemical gradient. When the hydrogen ions move through ATP synthase, the potential energy is converted to mechanical to twist the rotor and stalk. This is then converted to chemical to add the phosphate group onto ADP, regenerating ATP. This is called chemiosmosis. There's an alternate path which can occur without NADP plus or H2O. This is called cyclic electron transport. It only uses photosystem I, the cytochrome complex, and ATP synthase. To begin, a photon must be absorbed by photosystem I. exciting an electron of P700, which is now denoted as P700 star. This is then taken by the primary electron acceptor. The pigment now has a positive charge and is denoted as P700+. The electron is then transferred to ferredoxin, moving across to arrive at plastoquinone. A proton is moved from the stroma to the lumen by plastoquinone, which is then oxidized by the cytochrome complex. Finally, the electron is given plastocyanin and then photosystem 1, which is now neutral again. This process created a proton gradient, which helps to generate ATP through ATP synthase. This is essential to the Calvin cycle, which requires more ATP than NADPH.
Now, let's talk about the Calvin cycle and another super cool plant adaptation, C4 photosynthesis. By the end of this, you'll see how plants are basically little green geniuses. Let's get into it. No sunlight required. The Calvin cycle, also called the light-independent reaction or the dark reaction, is the second major stage of photosynthesis. It takes place in the stroma of the chloroplast and doesn't require light directly. Instead, it uses ATP and NADPH made during the light-dependent reactions. Its job is to take carbon dioxide from the atmosphere and turn it into molecules like glucose, which the plant can use for energy and growth. The Calvin cycle is made up of three main phases: carbon fixation, reduction, and regeneration of Ruby P. Each turn of the cycle fixes one molecule of carbon dioxide. It takes 3 turns to make one molecule of G3P, and 2 G3Ps are needed to make one full glucose. Let's break it down a little further. The first step is carbon fixation, where CO2 enters and binds to Ruby P, a 5-carbon compound. This reaction is catalyzed by rubisco, the most abundant enzyme on Earth. The 6-carbon product is unstable, so it immediately splits into 2 molecules of 3PG. In the Calvin cycle, this happens to 3 Ruby P for a total of 6 3PG. Now moving on to step 2, reduction. This is when the 3PG is phosphorylated by ATP and is turned into 1,3-biphosphoglycerate. But the goal is to make G3P, so NADPH donates an electron. This process happens to all 3PG molecules, creating 6 G3P molecules total. Of the 6 G3P molecules made from 3 turns, only one exits the cycle to help make glucose. This process is called gluconeogenesis. But now we have 5 leftover G3P molecules. What do we do with them? Step 3. Regeneration of Ruby P. The remaining 5 G3P molecules go through a series of reactions that regenerate Ruby P using more ATP. This keeps the cycle going so that the plant can continue producing sugars. So, what do we get from all this? To make one G3P molecule, the Calvin cycle uses 3 carbon dioxide, 9 ATP, and 6 NADPH. When we combine all 3 steps, we get the entire Calvin cycle. Once again, the 3 steps of the Calvin cycle is carbon fixation, reduction, and finally, regeneration of Ruby P. This process is crucial for life on Earth. It's how plants store solar energy in chemical bonds and lock atmospheric carbon into living systems. In hotter, more tropical conditions, plants close their stomata to prevent water loss. But this also blocks CO2 from entering, while oxygen builds up inside the leaf. Here's the issue. Rubisco can't tell the difference between carbon dioxide and oxygen. If it grabs oxygen instead, the plant ends up doing photorespiration, a process that uses energy but doesn't produce sugar. It's like running on a treadmill, a lot of effort, but you end up in the same place. Some plants evolve to work around photorespiration, like C4 plants, like corn and sugarcane. They separate the early stages of photosynthesis by location. In mesophyll cells, they use PEP carboxylase to fix carbon dioxide into a 4-carbon compound. This enzyme only accepts CO2 and not O2. Oxaloacetate turns into malate and then is transported into the bundle sheath cells. In these cells, malate is decarboxylated and then turned into pyruvate. During this process, we get carbon dioxide as a result, which is then used in the Calvin cycle, which is optimal for these plants. To wrap it all up, The Calvin cycle uses ATP and NADPH to convert carbon dioxide into sugars. C4 plants avoid photorespiration by separating carbon dioxide in order to capture sugar production by the cell type. These adaptations allow plants to survive in harsh conditions, proving once again that nature always finds a way. Thank you for watching. I hope you enjoyed learning about photosynthesis in different plants. Photosynthesis isn't just a chapter in our textbooks, it's the foundation of life on Earth. Thank you.